express my deep gratitude that the keynote address will be delivered by an extraordinary executive of our industry, and I deem him extraordinary for one simple reason. He's the only executive who has launched 2 CAR T cell therapies in the industry that I know of, the Yescarta and the Tiscartus, when he was the executive vice president at Kite Pharma, which is now part of Gilead. Many of you know Tim from the days of Genentech, where he served for more than 12 years as the senior vice president of technical operations globally. I recall there were 7,500 employees, professionals under him. He managed over 40 CMOs in 10, 12 different countries in the world. And I cannot think of anyone more entrepreneurial who can deploy technology to bring about the best in healthcare. So with that very brief introduction, Mr. Moore, the stage is all yours, and I know you will not screw it up the way I did.
Thank you, Devendra. Let me see if I can share, because it is coming up a little bit strange. All right. Can you see my screen?
Yes, sir.
Yes.
As you all know, Timothy is currently the president and chief operations officer of a company called PACT.
Yeah. So when I left Kite, which I didn't leave Kite because I wasn't happy. I left Kite because I was commuting back and forth from where I lived in San Francisco to LA every week. And my family finally said, "When are you coming home?" And I decided I did need to get back to family. And when I saw PACT as a company and what they were doing, I thought, wow, this is very, very cool stuff in cell therapy, because Kite has done such a great job and still is doing a great job, that I am highly motivated by the cell and gene therapy industry for advancing technology and innovation. So I want to share with you today is what PACT is doing, but also as an industry, what do I see we need to continue to do to help advance cell and gene therapy over the coming years. So to that end, PACT's vision is looking at curative T-cell therapies for cancers tailored to each and every patient. And what that means is we are focused on solid tumors specifically. And go to the next slide. In solid tumors make up about 90% of the cancer that's out there. And each person's cancer is personal and unique because there's highly specific mutations. And that's what limits treatment out there, that not all patients, let's say even for Genentech made monoclonal and still does make monoclonal antibodies. That's going after a specific target for that cancer disease, but not every patient will have that target and not at the same level. So what we're focused on is the neoepitopes, which are peptides originating from the tumor-specific mutation that binds to the patient's HLA. And then we make this neoepitope HLA construct and specifically gene edit that into their T cells. And to give you a sense of, again, how rare it is, less than 1% of neoepitope HLA targets are the same among individual cancers. That's what we've seen. So we're taking cell therapy to another level of personalized medicine. And again, I'll use the example, whether it's Kite, Juno, Novartis's product that's out on the market, is that has a specific target, CD19, constructed, and that's what's targeting every patient's, in that case, blood diseases. In this case, we are looking at each individual one. And to jump into there, this is an overall view of the PACT approach to developing this personalized T-cell therapy. And what I'll describe here is, in the blue box is where we identify and discover the neo-TCR and validate it's the right TCR. Then we make the patient's cell therapy. So to walk you through this is, while the patient is in a standard of care, we collect a tumor biopsy and blood sample from them. And what we do is we then do a bioinformatics beginning process. And in that process, we look at the prediction and prioritization of the tumor mutation by having it sequenced. And in that sequence, we look for that neo-E peptide. Once we've predicted and prioritized, we then generate a library, and we barcode these specific patient neo-epitope HLA proteins, because what we do with their blood sample is we want to capture in their blood sample these neo-HLA specific T cells from each patient's blood. And those barcodes, we call it our snares. And that way we can identify specifically which ones adapted to the patient. Then we clone them in step 4, and we functionally verify which neo-TCR looks like a strong candidate. And once we do that, we can select what we see up to 3 neo-TCRs. And we do that by looking at their functionality, the truncality, and also the diversity of those neo-epitope HLAs. And once we do that, let's say up to 3, we have a polyclonal product we're going to make for this patient. And that's when we then make, with the cloned TCR, we make a neo-TCR DNA plasmid. We have a manufacturing process using E. coli technology to make the targets. And then we trigger the patient, get their leukapheresis come in, and that's when we isolate the T cells, gene edit them, and that's what's called our Trifecta process. So the blue box is kind of the upfront that's going on while a patient's being treated with a normal standard care. And then once they progress, we're ready to edit their T cells just like in a CAR T type way, except we have different technology by immediately getting their leukapheresis. Now, to walk you through a little bit deeper, but I'll do it fairly quickly because our upstream process to discover and validate is designed to capture and verify those patients' polyclonal products. And this shows a funnel, because when you first get that biopsy sample and blood samples and you do the sequencing, you begin to see a lot in that. You can see thousands of potential targets. And from that, we begin to prioritize the mutation to then funnel down to see which barcoded patient-specific neo-HLA protein look the best. And that's where we take it down tenfold to in the hundreds. Then what we do is specifically to capture, we do a cloning epitope, and then we make this specifically for the T cells. And as I said earlier, put it in the patient blood samples. and see which ones functionally look the best. And that brings it down another tenfold to 10 potential targets to then when we pick those 3 targets. So this is a highly unique and innovative process to identify each patient's target. And to share with you what resonated with me when I first joined PACT is, if you think about this process with a little bit different mindset, like my days at Genentech, this is a research thing, because in research you identify targets, and once you identify a target, you then hand it over to process development to develop the process to make that target into a product. Well, what I'm trying to describe here is this will be a routine manufacturing process for every patient that we treat in this with this technology to really target each patient's specific tumor, even though they may have the same disease. So it's very unique, and we have a lot of IP along this technology. Then, as I said, we have a Trifecta process. We receive the leukopak, the leukapheresis from the patient, and the first thing we do, and we're using the Prodigy today, but we're looking at new technologies, is to isolate the CD4, CD8, and then enrich and activate those cells before. Sorry. Oh. Was there? Were you asking a question?
You're fine. Go ahead, Tim.
Okay. We activate, and then that prepares us to do what we, the gene editing, which we are using the technology of electroporation. So we aren't, our technology is not to gene edit viral vectors. We're using electroporation, which is the Cas9, also with the Cas9 technology. Then once we've gene edited the patient's T cells, we then expand those over, as you can see, about a 10-day period before we harvest the patient, and then obviously cryopreserve till the patient's ready for dosing. So to share with you what is unique, well, before I go to there, as you can see, this is, while the equipment has some automation to it, this is still a fairly manual process. Like a lot of the technologies out there in the cell therapy place, the unit operations, there's a lot of still manual handling of making the product in the suites. And what makes our cell therapy process so unique is we leverage this precision genome engineering technology that allows us to enhance the T cell functionality and trafficking. And specifically looking at the top, we knock out the endogenous TCRs and then specifically knock in our PACT neo-TCR. And what this does is it helps for a native normal functioning TCR, T cell, which is very, very important for the patient. Now, not to criticize viral vectors, what viral vectors do is they do a random knockout. So they're still fighting a little bit with the endogenous TCRs out there, so a little less functioning. In our technology, we're still advancing it as we are growing our platforms. The other unique thing about our process is we've designed it to increase the activity and persistence of the T cells by focusing on growing out the young phenotypes. As you can see in the top part 5 different types of T-cell evolutions. We are focused on growing out the edited cells that are the memory stem cells, central memory stem cells. It's not to say that we don't have effector memory and effector, we do, but we really focus on the majority of them being that. And we do that by we've optimized our growth media, and we don't have to use an IL-2. And this really helps us get to increased persistence in vivo, as well as a robust engraftment. Unlike, again, in my past, rapid expansion, it can happen very good with IL-2. But what that does is that does result in you're going to have more effector cells than the younger cells. And effector cells still are effective at attacking and killing tumor cells. However, there's a shorter lifespan in vivo. And so that's why we're focused on the young phenotypes. So I just wanted to share with you 2 of these very unique and innovative parts of our cell manufacturing process, as well as what we call our discovery and verification of identifying the targets in our process. So what does that mean for cell therapy product as you're wanting to grow not only clinically, but head towards commercialization? And it does pose a lot of unique challenges, but opportunities in the autologous cell therapy commercialization process. And if I start off with the manufacturing, as I said, there is complexity and it's resource intensive for manufacturing technology. We all in the cell therapy, not just about PACT, the whole industry is dealing with that. And while there is advancement in automation manufacturing to, lower cost of goods, help with scalability and reduce the amount of manpower you need to meet patient needs, this is still something the industry, I see, we need to continue to work as an industry team to help advance the companies out there working on automation and how to optimize that automation. It's made tremendous progress over the last 3 to 4 years, but it's not something I see is done. We got to continue to work, and I'll speak a little bit more to that in upcoming slides. The other thing we all know is about each patient is a unique batch in autologous cell therapy. And this requires chain of identity and chain of custody. And timing and scheduling are critical because typically, especially at the time at KEYNOTE, when you get a patient's leukapheresis, this is generally a patient that's not only progressed, is in a deep health problem. So you want to turn around as quickly as possible, getting that product back to them. And a lot of that in your early stage of clinical, you don't have quite the automation of the tracking and trending. And now there's systems out there like Tcell, Benetti, others that have optimized the business systems that you can track and trend and move things along. But this is something that I see still needs to advance. And one of those advancements, which I speak here in opportunities, is as an industry, we drive to standardization of those processes for tracking and trending, chain of custody and chain of identity. Because as you deal with the different clinics out there, they all can have different ways they're monitoring the patient's individual tracking and trending, and bringing that to a standardized that we all can optimize that, I think is going to be something in the future that will happen and will make a difference. And just to share with you our particular process here at PACT, since we are also having to track and trend each patient's tumor blood sample coming in where we're identifying the target. That adds another level of complexity to the process that the industry will need to advance on. And also, as you look at individual patients and you're gathering data end to end, having predictive analytic ways of monitoring and tracking and trending is going to be an important thing. And again, there's tools out there, but this is an area that still needs to advance. I've also seen in the cell therapy raw material, a lot of raw materials you may be using are from single suppliers. And I don't know about everybody, but during the pandemic environment, that could become a challenge where there be shortages on certain amounts of material. And so another thing that as an industry in the cell therapy, we really need to develop collaborative partnerships with our vendors to increase reliability of those raw material supplies. I can say at PACT, we worked our way through it during the pandemic, and now it's still, pandemic's going on still, but this is something that even once the pandemic lifts, it's still going to be an important thing because of the uniqueness of the raw materials in the cell and gene therapy space. The fourth area that is very important in this is the variability in incoming patient cells. Definitely is one of the challenging aspects of our manufacturing process to keep it consistent. Because we know each patient, depending on how their health is at the time, or just the type of DNA person they are, and they can have different cell viabilities coming into you.
Can you hear me?
Yeah. Was there a question? I guess not. And this is where I feel the opportunity is we need to continue to generate sufficient knowledge base on the apheresis material coming in from different patients so that we can optimize the industry in being able to process patients. An example which hasn't been implemented but is on my mind is you could have two different patient cells and you find out different medias work better in each one of those patients. How do you develop a GMP process that our regulatory world will accept, FDA, EMA, where based on the incoming patient's cell health, let's say, you use different a little bit different maybe raw materials or a little bit different technologies. It isn't like you're having to file an amendment. It's just part of the process. This is something in this space I think has to be thought through and worked with the agencies a little bit more as we move forward. And of course, the last one I wanted to point out was continue to look at ways to reduce adverse events to be minimized, and then we can optimize getting to more and more patients. Because in the industry today, and it has improved, when the CAR T therapies created some safety challenges, patients obviously stay once they're dosed, stay in the hospital for a minimum of 7 days, but it could be longer. And what that creates is it isolates to less and less hospitals you can treat in. And like in the industry, there's 100+ clinical facilities out there, but there's also commercial facilities just in different parts of the let's just use the US, the country, that patients may have to travel 3 or 4 hours to some place to get to the clinical facility because their local community facility can't do this process. Now, as we advance cell therapy and we get to where the risk of them having to be hospitalized, kind of like today, chemotherapy can just be given as a come in, get chemo, and then wait a couple hours and you go home. This is something that would really advance our capabilities to treat more and more patients routinely. So I just wanted to share these type of challenges and industry opportunities. Now here at PACT, oh, that's interesting, that didn't come up. Let me see if I hit down again, if it comes up. Oh, there it goes. Here at PACT, we're also looking at how would we scale out our manufacturing? Where are we today? We're definitely centralized, where that discovery and verification is all done in our same place as we're doing the cell therapy place. As we move closer and closer to commercialization, specifically in the US, we see that we'll expand, and again, like Kite's done, we'll expand in the US so that we can have manufacturing facilities closer to all the clinical. And we see that as being a very important thing. You know, you have one on the West Coast, you want one on the East Coast, one in the Midwest, so you can be very more efficient in processing versus you're shipping stuff all over. To that end, the upstream, where I talk about the discovery, we're saying we could keep that centralized because that technology is so unique that we could. But looking at the right-hand side, as you expand into more countries, Europe, Asia, I believe you'll have to decentralize and move across. Now, we know in the cell therapy industry too, there's a lot of talk about can we optimize our process to put it right into the hospital itself? And while that's in my mind, a very interesting and unique thought to think on, I think that's quite a ways away because of not only the manufacturing technology and where it's at today, but when I think about also how to incorporate all that from a regulatory GMP standpoint into hospitals. And then if you make a process improvement, you're in 100 hospitals, how do you do 100 tech transfers in an efficient way? Could be quite challenging, but I'm not saying it isn't something that shouldn't be considered over time. I think we're still at an early phase for that type of thing. And then as we're scaling our capabilities, these are some of the things at PACT that we are focused on and thinking about from clinical to commercial. The first one being is we have brought all of our discovery and manufacturing in-house, and we've done that because of continuous improvement. Is when you're in the early phase of any study, before you get to pivotal, you are always from your clinical study, but your manufacturing runs, monitoring patient incoming leukopaks, you're learning a lot. And here at PACT, we have learned a lot already and made process improvements to our process, which have been very positive in moving us forward. But then looking at the right-hand side and thinking forward, how do we scale out? And we are looking at designing, we're in a design phase, not in a build out phase of what would it look like to do commercialization. We are also partnering with companies out there to look at advancing the automation and technology. And just to share a couple things with you, the bottom picture you see there is that's our part of our discovery and verification upstream process that is being automated. Because think about, as I described in that funnel, you're getting thousands of patients' biopsies and you're discovering thousands to hundreds to tens of TCR targets. You're processing a lot of samples and a lot of devices. So we are looking at how do you fully enclosed and fully automate that, and we've got a path we're moving down. In the middle picture, this is for the cell therapy part. We're Solaris, for those of you that may not be aware of who Solaris is, they're a startup company that is in the process of developing not only their own CDMO manufacturing capabilities, but they're heavily automating, completely automating using also robotics technology. And we are partnering with them to look at how to do that because they are developing systems where you can significantly reduce the amount of handling. And today in the cell therapy, as we know, there's a lot of times you gotta, attach a lot of piping, of course, to do it in a sterile way, whether inside a hood or not. And Solaris is really developing, and it's still early stage, not to say it's at a GMP stage yet, but they're developing a very advanced technology that looks very promising for truly, as I said earlier, automating it to where we get more cost effective, more efficient in turnaround time for the patient's cells. So to summarize, the cell therapy industry is advancing, I feel, very well. But as it advances very well and discovering more and more ways to help patients not only extend their lives, but cure, I feel this is an industry that's going to need to highly advance the technology and innovation to make a difference. And I wanted to share with you some of the things we have going on and where I see more and more things needing to happen. So I will stop sharing there.
Tim, thank you so very much for giving us such a comprehensive insight into such a complicated process.
It is.
Thank you for the education. And equally importantly, if I didn't miss them, you gave us at least 6 areas, I'm going to call them missionary areas, for us to focus on as an industry so that we can advance the technology, the science, the operations, all the way from standardizing, tracking, and tracing to scaling out and enabling technology transfer. There are a bunch of questions. There's one I definitely would like to ask you before I let you go. What is the timeline for this therapy of yours to become commercially available?
Yeah, and as I showed on the one slide, the blue box and the green box, our timing where we're really focused on scaling it down is on the green box, because the blue box is going to be done while the patient's getting an existing treatment. So that's kind of in parallel. It's not an impact on the patient. The green part, which is the cell therapy part, we are working towards bringing that down to from the time we receive the apheresis from the patient to the time we would release to around, let's say, in the range of 25 days or lower.
And there are many issues you raised, which we will address during the course of this event. So thank you again, and hats off to you. Congratulations for the entrepreneurial work you're doing. Thank you, Tim.
Thank you.